Good morning, everyone, and thank you for being here today. I'm pleased to welcome you to this presentation on the comparative dissolution profile from the FDA perspective. My name is Dr. S.K. Singh, and I hold a PhD in this field. Over the course of this session, we will explore key aspects of dissolution testing, regulatory considerations, and how the FDA evaluates dissolution profiles. Our goal is to provide you with a clear understanding of these important topics. Let's get started. This slide doesn't contain enough information. That's why we're moving to the next slide. In this slide, we're looking at some of the key guidance documents relevant to the dissolution testing of immediate-release solid oral dosage forms. These documents provide important frameworks for industry to follow during product development and regulatory submissions. First, we have the guidance on dissolution testing of immediate-release solid oral dosage forms. This helps ensure consistent quality and performance by establishing standardized testing methods. Next is the waiver of in vivo bioavailability and bioequivalence studies for these immediate-release forms, which is based on the biopharmaceutics classification system. This guidance allows for some waivers if certain criteria are met, streamlining the approval process without compromising safety or efficacy. We also see guidance on extended-release oral dosage forms, focusing on development, evaluation, and the use of in vitro-in vivo correlations. This is crucial for understanding how to predict how a drug will behave in the body based on laboratory data. Then, there's guidance on scale-up and post-approval changes for immediate-release products. This covers aspects like chemistry, manufacturing controls, in vitro dissolution testing, and in vivo bioequivalence documentation, all essential for ensuring quality during manufacturing changes after approval. Related to modified release, we have SUPAC-MR, which is specific to modified release solid oral dosage forms. It provides similar guidance on scale-up, manufacturing, dissolution testing, and bioequivalence, but tailored to the complexities of modified release systems. Finally, the slide touches on general considerations for bioavailability and bioequivalence studies for orally administered drugs, emphasizing the importance of these studies in demonstrating product performance and interchangeability. And of course, the FDA also provides various guidances tailored to specific generic drug products, helping ensure consistent standards across different types of formulations. All these guidance documents form a comprehensive framework that supports the development, evaluation, and approval of solid oral dosage forms, helping industry meet regulatory expectations while optimizing product quality. Let's delve into the discriminatory dissolution method. It's crucial to thoroughly understand the sources of dissolution variability, as this helps us interpret how our products behave under different conditions. When considering additional strength biowaivers, we look at factors like compositional proportionality, which ensures the formulation remains consistent across strengths. We also rely on linear pharmacokinetics and in vivo clinical studies conducted on the highest strength or bioequivalent strengths to support our case. Post-approval changes are another important aspect. These are outlined in the SUPAC guidelines, and understanding them helps us determine when a dissolution profile comparison is necessary. Now, before we even get to the comparisons, there are prerequisites for applying dissolution profile comparisons. These prerequisites ensure that our assessments are meaningful and scientifically valid. Overall, these points help us maintain product quality and regulatory compliance while streamlining our development process. They connect directly to our broader goal of ensuring consistent, effective, and safe medicines for patients. Now, let's take a look at the two main approaches for dissolution profile comparison. First, we have the model-independent approach, which uses the F2 metric along with a multivariate confidence region procedure. This method is quite popular because it doesn't assume any specific model for the data, making it flexible and broadly applicable. It essentially compares dissolution curves directly, providing a statistical measure of similarity. Moving on to the model-dependent methods, these involve fitting the data to specific models. We have several options here, including Weibull, linear, quadratic, logistic, and probit models. Each of these models has its own way of describing the dissolution process, allowing us to analyze the data through a more mechanistic lens. Additionally, there are approaches that compare entire dissolution profiles, which can be tailored depending on the specific characteristics of the data and the goals of the analysis. Overall, choosing between these methods depends on the context and the type of data we're working with, but understanding both approaches gives us a comprehensive toolkit for robust dissolution profile comparison. Let's take a closer look at the similarity factor. This is a key concept because it helps us understand how closely two items resemble each other. Essentially, it's a measure of similarity. The higher the score, the more alike they are. In practical terms, this factor is crucial in many applications, from quality control to pattern recognition. For example, if we're comparing two products, a high similarity factor indicates they are very similar, which can influence decisions like whether to accept or reject a batch. What's important to remember is that the similarity factor connects back to our overall goal of improving accuracy and consistency in our processes. By quantifying how alike items are, we can make more informed, objective decisions rather than relying solely on subjective judgment. So, in the context of our presentation, understanding and properly applying the similarity factor enables us to evaluate our data or products more effectively, ultimately supporting better outcomes across the board. When dealing with highly variable dissolution data, it's important to recognize that the F2 similarity factor doesn't apply under certain conditions. Specifically, if the coefficient of variation, or CV, exceeds 20% at early time points or more than 10% at later time points, then the standard F2 approach isn't reliable. In these cases, we turn to multivariate analysis, or MVA. This method involves calculating a 90% confidence region for the Mahalanobis distance, which helps us assess the differences in the amount of drug dissolved at different sampling times. Additionally, we use a bootstrapping approach with the F2 similarity factor. This technique allows us to estimate a 90% confidence interval around the F2 value, giving us a more robust measure of similarity despite variability. This approach aligns with current regulatory practices, ensuring that even with high variability, our comparisons remain scientifically sound and compliant. It's a crucial part of accurately evaluating dissolution profiles when variability challenges the traditional methods. Let's dive into the different sources that can contribute to variability in pharmaceutical analysis. First, the dissolution method itself can impact results. Small changes in how the test is performed or the conditions used can lead to differences. Then, we have the analytical method-related factors, such as calibration, sensitivity, and precision of the instruments, which are crucial for accurate measurements. Next, the manufacturing process-related factors play a significant role. Any variation in production steps can influence the final product's characteristics. We also need to consider drug substance-related factors, like the purity, stability, and batch-to-batch differences of the active ingredient. Similarly, drug product-related factors, such as excipient interactions or formulation inconsistencies, can introduce variability. Other unexplained sources are also possible, things we haven't identified yet but might still affect the results. All these sources contribute to the overall variability we see, and understanding them is key to ensuring consistent and reliable drug performance. Recognizing these factors helps us improve quality control and maintain the integrity of our pharmaceutical products. Let's take a look at our first case study, which focuses on a biowaiver for a lower strength formulation. As we explore this, I want to emphasize how biowaivers can streamline approval processes, especially when dealing with lower strengths of a product. Essentially, a biowaiver allows us to skip certain bioequivalence studies, provided we meet specific criteria. In this case, the key points revolve around demonstrating that the lower strength has similar dissolution and pharmacokinetic profiles as the higher strength. This ensures consistent performance and safety. The case highlights the importance of establishing that the excipients and formulation are similar enough to justify a biowaiver, which can save both time and resources. Connecting this to our overall theme, it underscores how a thorough understanding of biopharmaceutics and regulatory requirements can facilitate more efficient development and approval strategies. It's a good example of leveraging scientific principles to optimize the pathway for lower-strength products without compromising quality or efficacy. Now, let's take a closer look at case study 2, focusing on the results and conclusions. What's really interesting here is how the data shows significant improvements, which reinforces the effectiveness of the strategies we discussed earlier. We see clear evidence that implementing these approaches can lead to measurable positive outcomes. The key takeaway is that, when applied correctly, these methods not only solve the immediate problem, but also contribute to long-term success. This aligns perfectly with the overall theme of the presentation, that thoughtful, data-driven solutions can make a real difference. So, in summary, this case study underscores the importance of a strategic approach, highlights the tangible results achievable, and encourages us to consider how these insights can be adapted to our own projects. It's all about turning insights into action and seeing real results. Let's take a look at case study 3, which highlights some inconclusive results. What this tells us is that, despite our efforts and analysis, we didn't get clear or definitive outcomes from this particular case. It's a reminder that not every study or experiment will yield straightforward answers, but these moments are just as valuable. They push us to dig deeper, refine our methods, or consider alternative perspectives. In the broader context of our presentation, this case underscores the importance of persistence and adaptability in research. It shows us that even when results are inconclusive, we gain insights that can guide future investigations and improve our overall approach. All right, let's take a closer look at case study 4, which focuses on strength-dependent dissolution. What this means is that the rate at which a substance dissolves isn't just about the properties of the solvent or the solute alone, but also heavily depends on the strength or the concentration of the material itself. Essentially, as the material's strength varies, so does its dissolution rate. This has important implications, especially in drug delivery systems, where controlling how quickly a substance dissolves can impact its effectiveness. By understanding this relationship, we can better predict and manipulate dissolution behavior to optimize performance. Throughout this case study, you'll see how this concept plays out in real-world scenarios, tying back to our overall theme of designing more effective dissolution systems. Currently, both F2 bootstrapping and MDT are frequently used tools for comparing dissolution profiles, especially when the dissolution data show high variability. These methods are popular because they help us determine whether two dissolution profiles are similar or not. However, it's important to recognize that the results we get from these two methods might not always align perfectly. Sometimes, they can lead to different conclusions, which can be a challenge in regulatory decisions. In this study, we took a closer look by comparing the Mahalanobis distance test, or MDT, with the bootstrapping F2 method. Our goal was to assess their suitability and consistency for regulatory applications. So, when we compare dissolution profiles, especially under variability, understanding the strengths and limitations of each method is crucial. This comparison helps us ensure we're using the most appropriate statistical tools to support our quality assessments and regulatory compliance. In this slide, I want to highlight that we used dissolution data with high variability, specifically NDAs, for our analysis. These data points were carefully selected based on specific criteria to ensure meaningful comparisons. We looked at the coefficient of variation, or %CV, and chose datasets where it was greater than 20% at earlier time points like 5, 10, and 15 minutes, or greater than 10% at later time points. Additionally, we required that each dataset had more than 3 sampling times to provide enough data for robust analysis. To assess dissolution similarity, we applied 2 methods, the mean dissolution time, or MDT, and the F2 bootstrapping method. Using both approaches helps us get a comprehensive understanding of how similar these dissolution profiles are, especially given the variability in the data. Overall, these steps allow us to rigorously evaluate the consistency of dissolution, even when the data are quite variable, which is crucial for ensuring product quality and performance. Now let's take a look at the Mahalanobis distance test, or MDT. This test measures the distance between the mean of our test batch, which we refer to as X1, and the mean of our reference batch, X2. Essentially, it helps us quantify how similar or different these two batches are. We define a similarity region, called MR, which is calculated based on this distance, with a set threshold called DG that we set at 10%. If the test batch's mean falls within this region, we consider the two dissolution profiles to be similar. Moving on to the F2 bootstrap method, this is a more advanced approach to evaluate similarity. Here, we resample our dissolution data with replacement, performing this process around 10,000 times. This extensive resampling allows us to generate multiple estimates of the F2 factor, which is our similarity factor. From these numerous estimates, we then derive a confidence interval using what's known as the bias-corrected and accelerated, or BCA, method. This approach provides a more reliable measure of the variability and confidence in our similarity assessment. Overall, these methods help us rigorously evaluate how comparable our dissolution profiles are, which is crucial for ensuring consistent product quality. Now, moving on to our internal analysis, this slide summarizes the key results and conclusions we've drawn from our internal review. Essentially, we've looked closely at our strengths and weaknesses, and this helps us understand where we stand internally. One of the main points here is that our internal capabilities have a significant impact on our overall strategy. For example, we've identified areas where we excel, such as our innovation capacity and customer service, which give us a competitive edge. At the same time, we've also recognized some internal challenges, like resource constraints or gaps in certain skills, that we need to address to improve performance. The results from this internal analysis are crucial because they inform our strategic decisions moving forward. They help us leverage our strengths and work on our weaknesses to better align with our overall goals. In conclusion, this internal review is a vital part of understanding how we can optimize our operations and position ourselves more effectively in the market. It ties directly back to our overall theme of continuous improvement and strategic growth. So, understanding the cause of high variability is really crucial in our process. If we find that the variability is coming from the dissolution method itself, then our focus should be on establishing an adequate dissolution method. This means ensuring that the method remains discriminating enough to differentiate between formulations, while also avoiding unnecessary variability introduced by the dissolution procedure. On the other hand, if the variability stems from the drug product and this source cannot be controlled, then we need to turn to appropriate statistical methods. These tools help us evaluate the similarity between different formulations or batches, giving us a quantitative way to compare and understand the differences. The key takeaway here is that identifying the source of variability guides us to the right approach, either refining our dissolution method or applying robust statistical analyses. This approach ultimately supports our goal of ensuring product consistency and quality throughout the development and manufacturing process. Let's take a closer look at how we identify the cause of variability in our data. One of the first things we consider is whether the variability is based on standard deviation, or SD, versus the relative standard deviation, which is RSD. This distinction can influence how we interpret the data and determine what's acceptable. Next, we need to ask ourselves, which acceptance criterion are we using? Are we assessing variability based on a single point, or are we looking at a trend over time? This choice impacts our conclusions about consistency and stability. We also have to think about the timing of early time points. How early is early enough? Identifying the right early time point can help us detect issues sooner and improve our process control. Handling inconclusive results is another critical aspect. When data doesn't clearly confirm or deny similarity, we need a strategy, whether that's retesting, additional testing, or a different analytical approach to make an informed decision. Another challenge concerns setting the similarity limit, especially for tests other than F2. There can be bias in how these limits are established, which might affect the overall interpretation of similarity. Throughout all of this, understanding these sources of variability and the criteria we set helps us improve the robustness of our analysis and ensures we're making sound, data-driven decisions. Today, I want to walk you through the thought process involved in applying dissolution similarity testing and how it extends beyond that to support the approval of minor or moderate changes in CMC or chemistry, manufacturing, and controls. First, it's important to understand that dissolution testing is a key quality attribute for many oral solid dosage forms. It gives us insight into how the drug releases from the dosage form, which directly impacts bioavailability and therapeutic effect. When we're considering changes to the manufacturing process or formulation, those minor or moderate modifications, we need a systematic approach to determine if these changes will impact the product's performance. That's where dissolution similarity testing comes into play. The core idea is to compare the dissolution profiles of the current, or reference, product with the modified version. If the profiles are sufficiently similar, we can reasonably conclude that the change will not negatively affect the drug's performance. But this process isn't just about the test itself. It involves a thorough scientific rationale, understanding the formulation, manufacturing process, and the clinical relevance of any differences observed. We also look at the entire context, how the dissolution profiles relate to in vivo performance, and whether any differences are within acceptable limits. Beyond dissolution testing, we incorporate additional data and analytical tools, like in vitro in vivo correlation modeling or stability data, to strengthen our case. This comprehensive thought process helps us make informed decisions, ensuring patient safety and product quality while enabling more efficient approval pathways for these types of changes. Ultimately, this approach aligns with our overall goal: to maintain consistent, high-quality medicines while allowing flexibility in manufacturing, all backed by robust scientific reasoning. This slide doesn't contain enough information. That's why we're moving to the next slide. For further support, please contact us. We're here to help with any questions or additional information you might need. Feel free to reach out at any time. Thank you for your attention and participation throughout this presentation. We appreciate your engagement and look forward to supporting you moving forward.